Hello, everyone. Welcome to this webinar today. I'm Kenny from ATCG Limited, who is the distributor of IVIM technology in Hong Kong and Macau markets. In today's session, we will be discussing the real-time intravital microscope in vivo cellular level imaging of internal organ in a live animal. We will explore how advanced two-photon and confocal microscope enable high resolution in vivo imaging and vivo cellular dynamic across organs with application in drug delivery and disease research. Now I'm pleased to introduce our presenter today, Dr. Tim Dr. Tim from IVM Technology. Dr. Tim earned his degree in electronic engineering from Korea National University, and he does his postdoc research in and completes his postdoctoral research in Harvard Medical School. He's veteran leader in vivo cellular imaging, focused on advancing intravital, confocal, and two-photon microscope to enable real-time visualization of disease process and support the development of new therapies. Please don't hesitate to submit your question in the Q&A box at any time during the presentation. We will do our best to answer as many as possible during the Q&A session at the end. So, Dr. Tim, if you're ready, please go ahead with your presentation. Many thanks.
Okay, so thank you. Thank you everyone for joining today's webinar. And then thank you, Kenny, for the very detailed and kind introductions. Okay, so today I will talk about the real-time intravital microscopy technique, which is very useful for in vivo cellular-level imaging of various internal organs in a live animal. And then also it can be a very useful tool for imaging analysis, in vivo imaging analysis of drug delivery and drug efficacy in a live animal. Okay, so I have a CY to declare. Okay, so this is the data you can expect to get by using intravital microscopy in a live animal model. So compared to other imaging technologies such as X-ray, PET, CT, MRI, ultrasound, and optical imaging systems such as IBIS, the intravital microscopy allows us to do this microscale cellular level, video and imaging in a live animal model. So this particular example on the right-hand side, this is a 10-minute video obtained from the lung of a live anesthesia mouse model in vivo. So in this example, we are looking at the blood flow in green color, which can be visualized by intravenously injected FRC spectrum. So they stage inside the blood vessel. So we can see the blood flow. And then in red color, what we are looking at is the neutrophil moving inside the blood vessel. So this neutrophil is specifically labeled by antibody for conjugate. Those were again intravenously injected at 30 minutes before the imagings. So as you can see in this video, we can very nicely and very clearly see the individual cell movement inside the vessel in this lung in the live anesthesia mouse model. And then we can see how these neutrophils move around inside the vessel and how they do interact with each other, so making a cell cluster. And then eventually this cell cluster block the blood flow in here, in this area. So as you can see in this video. Okay, and this scale bar is 20 micrometer. So which I can say, this is a really microscopic scale imaging result. So in addition to that, so not just a cell, we can also see anything inside a live animal with presence labeling. So in this particular example, so we label the nanoparticle in red fluorophore, okay, in here, as you can see in here. And then we intravenously injected those presently labeled nanoparticle. So it's kind of drugs into the tail vein. And then we can take this real-time video from the liver in vivo. And then as you can see, we can nicely see how this intravenously injected nanoparticle appears inside the liver capillaries, liver sinusoid, and how they accumulated and delivered into the liver in real time. So in short, intravital microscopy is a very useful tool that can acquire these live cellular level videos, live animal model with some molecular information as well. And then also, as you can see in this example, it can be used to directly see the drugs in a live animal model. Okay. So by using our intravital microscopy system, we have been able to image almost every organ and tissue in a live animal model. So all these images were obtained from the live anesthetized mouse model in vivo. So we have been able to image the bone marrow, brain, this is brain tumor, again in a live mouse model, retina, skin, these are the hair follicle in the skin. And in various tumor model, this particular example is a colorectal tumor model, pancreas, spleen, or GI tract, adipose tissue, prostate, placenta, lymph node, memory tissue, and in various type of transplant. This is a cell transplant. So this green dot is the transplanted cell, kidney, muscle, liver, heart, lung, and so on. So we can image all these different organs with very high quality and in very high resolution in a live anesthetic mode, like in live mouse model. So our dual mode intravital confocal two-photon microscope looks like this. So it has a very compact size with a 63, 89, and 45 centimeter in width, length, and height. So this is the one very single box. And this is a very optimized system for in vivo, cellular level intravital imaging of live animal model, as you saw in the previous slides. And then this microscope can do both of two-photon imaging with 4 fluorescence channel, or at the same time, it can also do confocal imaging with 4 colors at the same time. So this microscope can do both of two-photon and confocal imaging. And then this microscope has very high-speed laser scanner inside for real-time imaging with a speed of 50 to 100 frames per second with 512 by 512 pixels per frame. So this is a very fast real-time imaging system. And based on this real-time imaging capability, we built in the automatic live tissue motion compensation function. So let me explain more about this automatic tissue motion compensations. So if you think about the live animal imaging in very high resolution, such as microscope, confocal and two-photon microscope, due to the heart beating and then breathing motion of a respiratory system, we always suffer this microscopic tissue motion, scan tissue motion. So this is the very typical tissue movement motion you can see when you look at the internal organ in microscopic scale, right? So we need to deal with this motion content, these motion issues to obtain the high quality images and videos. So, but our microscope is very fast, so it's capable of real-time video rate imaging with very high speed. So we can track individual cell movement inside this tissue, right? So like frame 1, frame 2, frame 3, and frame 4, we can track the its individual cell and then determine the tissue movement and the level of focusing. So later we can do frame by frame analysis and do the image registration to compensate the tissue motion and select the best image frames. And then this is the result. Okay. So from this kind of tissue motion, we can remove all the tissue motion and generate this very high quality images from the moving tissue in real time. Right? So without this motion compensation function, or with the slow microscope, you will always only obtain these very blurry images from the moving tissue. And then we made this motion compensation function to be automatic. So all you need to do is just turn on the motion compensation function and press the image capture button. Then in just a couple of seconds, you will get this highly precise motion compensated images from the moving tissue in a live animal model. Okay, in addition to that, this real-time imaging also allows us to do very precise denoising of the high-speed video. So this is actually published work in Nature Methods in collaboration with another professor at KAIST. And then the good thing, the beauty of this denoising algorithm is it doesn't need any pre-training, and then it's unsupervised method, so it can be done automatically. And then this is the best optical denoising algorithm for high-speed video. So it's a good match with our video rate imaging data obtained by using our microscope. So this is one example. I hope you can see this real-time video. So on the right-hand side, this is a low video, the very high-speed real-time video obtained from the skin of an NSIG mouse model. So in here, in blue color, you can see the flowing red blood cell inside this capillary in the skin. And then after denoising, after this automatic denoising, we can improve the signal to noise ratio more than hundreds of times. And then we can distinguish individual endocellular cells and the flowing red blood cells in real time in a live mouse model, as you can see in this example. So another example, so this we can also denoise many fast moving cells, denoise fast moving cells, as you can see in this example. So this is another video obtained from the muscle. So again, we can nicely see the flowing cells and then individual nuclei in their muscle fibers, right? And then even this very noisy videos can be very nicely denoised, as you can see in this example. This is the real-time video obtained from the kidney of a live NSIG mouse model. So if you compare the low video and the denoised video, you can nicely see the power and the beauty of this AI denoising algorithms. And another example, so this is the brain. Okay, so in this example, we are imaging this neuron, and then we can remove all the noise from this real-time video, and then clearly distinguish the individual dendrite of this neuron from this noisy real-time video. Okay, so in the example I showed you in the first slide, so this is how we prepare the mouse model for the intravital imaging of the liver. So mouse is here, and then we are imaging the liver, right? So actually, this was the real-time video, and then this is the denoised video. So even in the real-time video, the low video, we can still see the delivery of a single nanoparticle into the liver capillary. But after denoising, we can have a much better resolutions and then much better signal to noise ratios. So as you can see, so we can nicely distinguish the individual nanoparticle inside the blood vessel in the liver. Okay. So this real-time imaging and automated denoising software is now available for free trial. So and then it can deal with all different type of video formats. Okay, so if you are interested in using this software for your research, please ask us. Okay, and then as I told you, so our microscope is capable of the default imaging speed is 50 frames per second. But if we decrease the number of lines in our individual frames, then we can speed up the imaging speed even further, much up to even further up to 400 to 800 frames per second. So it can do ultra fast imaging video recording live animal model. Okay. So as I told you, so based on this real-time imaging capability and the automatic tissue motion compensation allowed us to obtain this beautiful live images from the live animal model in all these different organs. Okay, and then now let me show several examples of how we imaged all these different organs in a live animal model. So this is the example of the imaging of the lung in a live NSIG mouse model. Okay, so this is how we prepared our mouse model. So mouse is anesthetized, right? And then the mouse is intubated, and then we expose the lung, and then by taking out the air from this inside the imaging chamber through this suction hole, through this suction tube, we can build up the very gentle, negative air pressure inside, enough to stabilize this long surface to the transparent cover glass. So you can see in here. So now it's stabilized. So after stabilization, we apply the tissue motion compensation function, and then finally we obtained this very high resolution time-lapse video from the lung of a live anesthetic mouse model. So as I explained to you, we can distinguish individual neutrophil inside the vessel, and then how they move and how they interact, and then make a cluster, and then disturb the blood flow. So if you compare these images and these images, we can clearly see the disturbance of the blood flow in the lung due to these neutrophil interactions. So yeah, this is the procedure. So for fun, so first we need to intubate the anesthetic mouse model, as you can see in here. Okay. And then we connect this tube to the mechanical ventilator, so to keep the mouse to breathe air during this surgery. And then we expose the lung, stabilize it, and then we put we put this mouse model into our microscope using on this motorized 3D stages. Okay, like this, and then we make we find the focus, and then obtain the microscopic scale images. Okay, and then we can use the same principle for the beating heart imaging in the live mouse model. So again, the mouse is intubated, and then we use the same principle. So in here, we have this tube. So we suck out the air, build up the negative air pressure, and then stabilize the surface of the beating heart to the glass. And after this stabilization, we can obtain this real-time video of the heart vessel, the coronary vessel, and flowing cells, and then some immune cells in green colors. Okay, and then let me tell you, how long can we image the live mouse model? The answer is as long as we can keep the mouse to be anesthetized. So this is one example. This is an example of 2-hour imaging obtained from the lymph node in the live mouse model. So in this example, so again, the mouse is anesthetized, and then we catheterize the tail vein, so we can inject the cells through the tail vein intravenously. So we inject this red fluorescent T cell and green fluorescent B cell through the tail vein. And then we take a 2-hour video from this lymph node, and then observe the extravasation of this T cell and B cell in this HB, the high vessel venue, from the lumen of this vessel to the lymph node parenchyma. So if you look at it carefully, so we can see how these T cells and B cell get out from the lumen of the vessel to the lymph node parenchyma. So if you look at here, we can see this red T cell comes out very quickly. So again, if you look at here, we can see this green cell comes in, and it takes some time, but they eventually get out, right? So this kind of cell trafficking and then cell movement can be very nicely visualized by using our intravital microscope. Okay, and then I already show you this liver imaging, right? So let me go on. So that was just one example. So, and then another thing is, we can keep the mouse to be alive during the intravital imaging, but after each imaging sessions, we can recover the mouse from the anesthesia and then put it into cage. And at the later time point, we can bring the same mouse again and then image again. So we can do the repeated longitudinal imaging of the same mouse model. So in this particular example, we induce fatty liver disease, non-alcoholic fatty liver disease in the live mouse model using the MCD diet. And then with the MCD diet, this mouse build up the lipid droplet, inside the hepatocyte. And this this lipid droplet can be labeled by using some fluorophore called SF44 in vivo. So again, so we can see this gradual, longitudinal changes in the liver by using our intravital microscopy system from day 2 to day 21, and then we can see how this, yellow lipid droplet, accumulate over time. Okay. So another example, so not just lipid, we can also observe the cell, transplanted cell in the live mouse model for a very long time. So this is the example of the bone marrow imaging after bone marrow cell transplantations. So this is very wide area images obtained by using tile scan, mosaic functions. So this is day 1, day 3, and day 4. And then we repeatedly image the same mouse after the bone marrow cell transplantation. And this red color is the transplanted cell expressing RFP, DS-LED. And then at every day, we can find the exact same location, exactly same area inside the bone marrow by looking at the blood vessel in blue color here. And then we can find the exact same location, and then we can see how the transplanted cell appears inside the bone marrow, as you can see in here at day 3. And these cells, proliferate, right? Very rapidly proliferated until day 7. And suddenly, in just one day, these cells were immobilized and disappear from the bone marrow and go to the periphery. And then, interestingly, we can see this neighboring, site, the neighboring niche now have proliferating cells. Okay, so we can image the same mice, same area, at multiple time points, and observe how the transplanted cells changes, and then how the tissue changes over time. Okay, so of course our microscope can capture how these individual cells, this was low resolution imaging, how the individual cells behave inside the bone marrow. So this is a 5-hour time-lapse video to show the cell movement and their proliferations. So in white arrow, you can see this proliferation of the individual transplanted cell, and then in yellow color, we can see how these cells are collected into these areas. Okay, and this is the example of the small intestine imaging. So in case of small intestine imaging, we extrude the small portion of the small intestine and expose the lumen. And then in this particular example, we use proxon GFP mouse model to see the lymphatic vessel. And then, so this green color is the rachia. Those are the lymphatic vessel in the middle of the villi. And then red color is the fluorescent fatty acid. So we put it onto the small intestinal lumen, and then see how this fluorescence fatty acid can absorb into the villi, and they go into the laminar porphyria, and then transport this to this green lymphatic vessel, right, over 26 minutes. And then when you look at the mesenteric lymphatic vessel, this is in green color is the mesenteric lymphatic vessel, we can see how this fatty acid is absorbed into the villi and then transported to the lymphatic vessel in the mesenteries over time. Okay, so that was the few example, and then let me try to explain about the imaging window technique for longitudinal in vivo imaging of the same mouse model. So I like to discuss 3 techniques. The first one is the dorsal skin fold chamber technique. This is the very useful and a very well-established technique for cancer xenographic imaging. So this is the procedure. So we implant this metal frame into the dorsal skin of the live mouse model, right? So and then we suture this window into the dorsal skin, okay? And then we expose the subcutaneous tissue by removing some portion of the skin. And then in the middle, we can inject the fluorescent cancer cell into the subcutaneous tissues. Okay, and then we sealed up this dorsal skin fold chamber. And then after this surgery, we can keep the mouse to be alive to more than 3 to 4 weeks, and then repeat the imaging in the same mice again and again. So this is one example. So in this particular example, we inject this LIC-GFP cancer cell, which is in blue color, and then we label the blood vessel by using anti-CD31 antibody, intravenously injected anti-CD31 antibody conjugate with the fluorophore. So it nicely labeled endothelial in red color, and then we can directly see the flowing cell inside the tumor vessel. Okay, and then as I told you, after this, transplantation of implantation of dorsal skin fold chamber, we can keep the mouse to be alive for 3 to 4 weeks. So it's a very useful, technique to monitor the, drug delivery into this tumor model. So this is one of the collaboration work we have done. Okay. So our collaborator made some nanoparticle that can specifically target the activated tumor endothelium. Okay. So what we have done is we made this triple-negative breast cancer mouse xenograft model by using MDA-MB231 GFP cells. And then we let this breast cancer cell grows for 2 to 3 weeks. And then, so as you can see, we can build up the very nice tumor. And then by using our microscope, we can distinguish individual tumor cell inside the breast tumor, inside this dorsal skin fold chamber. And then we intravenously inject our nanoparticle through the tail vein, and then take an image at 2 hours, 6 hours, and 24 hours after the injection of the nanoparticle. So let me remove the green color. So as I told you, this nanoparticle specifically target the tumor endothelium. So at 2 hours and 6 hours, you can see this, you can see all of the nanoparticle inside the tumor endothelium, right? And then at 24 hours, if you look at here, we can see this nanoparticle get out from the vessel and then delivered to the like this. Okay, so 2 hours, 6 hours, 24 hours, and then we can see how this nanoparticle delivered to the single tumor cell. So this is just, again, this is the same mice model. So by just looking, this blue color is the tumor vessel. So by looking at the vessel, we can find the exact same location, and then we can repeat the imaging in the same mice model, and then see how how these injected nanoparticle delivered to the tumors. Okay. And then in this particular example, we take an image until 6 hours after the injection, and then we walk up the mouse model, put it into the cage, and the next day we bring the mouse again and take out, and then take an image again, and then find the same location, and then take these images at 24 hours. Okay. So that was a good example. So there was a very nice review paper in Nature Liver Cancer 2014. So how we can use the intravital microscope for the drug development. So as I showed you in the first place, we can use intravital imaging for visual profiling of the drugs, so to find the target, to discover the target. And after that, we can use the intravital imaging for drug delivery analysis, as I showed in previous example, and then also for the drug efficacy analysis. So let me introduce one example. So this is the work published in Science Advances at 2020. So in this example, we injected the fluorescently labeled exosome in red color through the tail vein. And then this is the before injection. This is 10 minutes and 30 minutes after the intravenous exosome injection. So if you look at this cell, so even just after 10 minutes from the tail vein injection, we can see the very rapid exosome targeting to this target cell in here. And then at 30 minutes, we can see that this exosome is now internalized into the cell. So this is a very good example of using intravital imaging for drug delivery monitoring. And another example, so in this case, we observe the brain for 48 hours after the injections. And in this case, this red color is actually antibody drugs. And then upper panel is control antibody drugs. And then lower panel is modified antibody drugs using brain shuttle, the BBB shuttle. So after modification, this bottom panel, these antibody drugs can penetrate the BBB in the brain. So if you compare at 24 hours, you can see the delivery of these antibody drugs to the brain tissue. But in contrast, the control antibody drug, they cannot penetrate the BBB, and then they only stay inside the blood vessel, and later it disappears, right, at 48 hours, if you can see here. But in modified, antibody drugs, they stay in the brain for more than 48 hours. So again, this is a very good example to monitor the drug delivery in a live mouse model, and then clearly demonstrate the efficacy of this antibody drugs modification in the live mouse model. So, and then let me show you some other example. So this was the first example, and then I already show you that this neutrophil clustering is the cause of this capillary blocks and the microcirculation dysfunctions. So this neutrophil can be our target, right? So this is another example, another observations, another experiment. So in this case, so this red color is actually RBC, red blood cell. So we don't see any RBC in this area, which means this microcirculation is disturbed in this area. But after this drug treatment, called abscissima, we can see the recovery, the reappearance of this RBC perfusion in this area. So it nicely, very clearly shows that these drugs can target the neutrophil and then restore the microcirculation in the lung. So this is one good example of how we can use intravital imaging for drug efficacy validation in the live mouse model. Okay, so another example. So this again, this is dorsal skin point chamber. So this green color is the tumor cell, and red color is the blood vessel. Again, in vivo labeled by CD31 antibody conjugate, flow conjugate. And then we can see how this vessel changes at 3 days interval. So we can find the exact same location, and then how the blood vessel changes over time with tumor cell growth, right? So if you look at here, without treatment, from day 7 to day 10, we can see the huge infiltration of this green tumor cell, and then subsequently, it make the tumor vessel to be enlarged, as you can see in here. So you can see the huge vessel dilations, which is due to the vascular growth factor, secreted from this tumor cell. So if we can target those growth factor with some anti-angiogenic treatment, we can strongly suppress this kind of vessel remodeling. So again, this is same mice model, day 7 to day 13. This is same mice, so same location. So we can see the very huge vessel remodeling without treatment. But with some anti-angiogenic treatment, if you compare day 7 to day 13, it didn't change a lot, right? So this anti-angiogenic treatment is very effective in suppressing this kind of vessel remodeling in this tumor model. So this is the before and after the treatment. So this is a very clear evidence of the efficacy of this treatment, this kind of a treatment. So another example, so the abdominal imaging window, which is a ring-shaped window that can implant onto the abdomen or side of the mouse model for the longitudinal repeated imaging of the abdominal organs such as liver, kidney, pancreas, spleen, and so on. So this is one example. So we implanted this abdominal imaging window on the side of the mouse model to see the spleen and the pancreas. And then when we implanted this imaging window, we inject the mouse pancreatic cancer cell, PANC2-GFP, here. And then we observed how this tumor cell grows and the tumor vessel changes up to several weeks. So this is from day 9 to day 14. So again, this is a single one mouse, and then we just repeat the imaging at different time point, and then find out the exact same location from day 9 to day 10. It doesn't change much. But if you look at here in this vessel, from day 10 to day 11, we can see the huge dilation of this vessel, right? And then very dynamic, angiogenic changes happen from day 11 to day 12 in here, right? But compared to that, if you look at this area, vessel initially looks like this, doesn't change, but now it grows. So we see some anti-angiogenic changes, but later the vessel regressed, right? So when we look at when we follow these tumor vessel changes with the growth of this pancreatic cancer cell, we can nicely see how these tumor cell and then tumor vessel interact with each other and then change their morphologies and functions over time in a single mouse model. Okay. Then let me introduce this cranial imaging window technique. So this is another very well established technique for the long-term, brain longitudinal imaging. So in here, we remove the small portion of the skull, so make a hole in here, and then expose the brain. And then we cover the exposed brain with the transparent cover glass. Okay, and then we protect the exposed skull with the dental cement. So as you can see, so we put the dental cement to protect the brain, the skull. And then through this window, we can nicely see the brain tissues. So after this surgery, mouse can survive more than 1 year. So we can keep the mouse alive for more than 1 year and then repeat these brain imagings. So this is one example. So after this surgery, we can very nicely see the blood vessel inside the brain cortex and the leptomeninges and dura. Okay, in here. In this particular example, we are looking at the pericytes in red color, and the endocellular cell in blue color, and green color is the blood flow. Okay. And then let me introduce one study using this cranial imaging window technique. There is one photothrombotic chemical called rosebenga. So in this example, so we intravenously inject the rosebenga, it's in blue color, and then we look at the platelet in green color. Okay, and then by using the laser beam, we can specifically make a clot, make a thrombus inside the vessel in here, as you can see in here, right? So we can target the single vessel and then make a thrombus inside, and then make a microinfarction. So what we can do is first we find out the penetrating arteriole in the brain cortex in here. So you can hear this is the penetrating arteriole, and then we inject the rosebenga, and then shine the laser beam, and then block the blood flow here, and then make a microinfarction. So after that, we repeat the imaging for up to 1 month. So this is the exact same location again, day 0, day 1, day 2, day 5. So in the beginning, we can see the expansion of tissue, such as it means we have edema, the tissue swelling, right? So tissue swelling until day 2, and then recovery around day 7, day 5, day 7, and then atrophy, the tissue shrinkages. Okay. So in the early phase, so this is day 0, day 3, and day 5. So if you look at this red color, this is 3000 Dalton dextran. Okay. At day 0, because there is BBB, there is no leakage. But at day 3, after the microinfarction, we can see this leakage of this red color 3000 Dalton dextrans. Okay. So that's why we have tissue swelling, edema at day 2 and day 3. But spontaneously, vessel doesn't leak out anymore at day 5. Okay. So this is the same vessel, same location. So vessel, recovered, their BBB functions, right? So we are wondering what's happening. So this is the 24-hour continuous, timeless images obtained from the brain after this microinfarction from day 3.5 to 4.5. Okay, and here, if you look at this red color here, okay, it elongates their processes along this blue color endocellular cell. Okay, so we can clearly see the process elongation of this pericyte along the endocellular cell, and then this is the major underlying cellular mechanism for this BBB function recovery from day 3 to day 5. Okay. And then also we can see the many other types of cell. And then, so this is one example. This is microglia. So we use CXACR1 GP mouse model. So at day 0, we can see this nice microglia, non-active, stable. At day 1, we can see the huge, activation like this one. Okay. So, microglia, change their shapes, and they became very active. And then we can also see some infiltrating monocyte, if you look at here, right? This small circular shaped cell is the monocyte. And then you can repeat the imaging at the same vessel, same area. So this is the exact same vessel. So at day 4, day 11, day 25. So it activated until day 4, and some activation until day 11, and then it get to normal again at day 25. So we can see the the microglia activation and then changes over time. Okay, and then this is another example. So using cranial imaging window in the middle, we can see this lymphatic vessel, the meningeal lymphatic vessel inside the mouse, in the mouse model. And then there is another technique called granular imaging window, which has these cannular shapes. So it can be inserted deep into the brain cortex. And then by using this technique, we can image the deeper brain areas such as hippocortex, hippocampus, cortical plexus, and corpus callosomes. So this is one example. In this area, in this particular example, we target the cortical plexus deep in the brain. So we remove the layer of the cortex and then see the hippocampus or choroidal plexus. And then this is the how the choroidal plexus vessel looks like through the this CIC, the cannular imaging window. And then we can also image the hippocampus and then see this GBM cells. So this is GBM TD tomato cells in red color, and then see the how the blood vessel changes for 3.5 days for 84 hours. So you can see this vessel gradually get expanded, get dilated in just 3 days. Okay. This is one example. Okay, so I showed you all these different examples of how we can images all these different organs, right? So again, so let me briefly explain about our microscope. So microscope is capable of four-color two-photon and four-color confocal imagings. So this is one example of the four-color imaging obtained in the skin of a live mouse model. Okay, so this green color is actually nuclei. Red color is the cell membrane. Rosa 22 is Rosa 26 empty. And then blue color is the blood vessel, actually blood flow. And the white color is the second homogenization signal from the collagen in the dermis. So again, another popular example. So all these images were obtained from the live and stage mouse model. So this is the muscle. So in the muscle, we can nicely see the collagen in the fascia in the beginning, and we can also see the sarcomere myofilament inside the myofiber, right? It has very fluid structures, and then we can distinguish individual nuclei. And then not just a single area. What it can do is, if we want to see the large area, wide area, we can take one image here, move, and then another image, another image, another image, like a TIE scan. And then later we can do automatic image stitchings to make this wide area mosaic images. So again, so this is the in vivo images of the confocal image of the pancreas, large area images. So again, this is kidney. Okay, we can see this very dense capillary network between the tubule, right? And then this is the large area images. Okay, and then also we can do both of two-photon and confocal imaging in the same mice model. So we can switch between two-photon and confocal imagings. So again, this is very wide area images obtained from the muscle. And then in here, in this example, this green color is peripheral nerve. And it's visualized by this Zion YFP. So we can nicely see, you can take a G-stain image, and then later we can build up the this very nice neuromuscular junction images from the live mouse model. And then if you look at here, also we have some heating system to keep the mouse body temperature to be 37 degrees during the imagings. So this is how it looks like. This is our tablet system. And then all our imaging system has a heating pad, heating system inside. So this plate for the mouse has a heater inside. So we can monitor the mouse body temperature through this rectal probe. Okay, and then we feedback control the temperature of this heating plate like this, and they keep the mouse body temperature to be 37 degrees Celsius during the imaging. Okay, so when you open our microscope, so we have this motorized stages. So and then we can put our mouse model on the plate heating system. And then we put it into the under the objective lens, and then take images. And then we can also connect in the gas sensitization system. So we have a two-tube adapter here. So we can connect the isoprene vaporizer system. So we can do anesthetization gas in, goes in, and goes out. Then we can keep the mouse to be anesthetized during the imagings. And then we have a several modified model, which we call FS system. It's the same system, but it has a much larger open space. So it's a good match for the larger animal images, such as acid lab. And then even from the acid lab, we can see all these different organs very nicely, as you can see in these examples. And then for each different organs, we have an optimized imaging windows and chambers for longitudinal in vivo imagings. Okay. And then also, so by using these techniques and solutions, we can image all these different organs. And then it's also important to have the nice software for the image processing and visualization. So although our microscope is intravital microscope, it's optimized for the in vivo imaging of the live mouse model, but it can also image the slides, standard slides, as you can see in here. So this is a very wide area imaging of this brain full slides. And this is actually more than 25,000 images are automatically stitched. So like this. So we can do very large 2D imagings. And then also we can deal with the live cell culture system and the organoid system. And then also, this software, which we call studio software, it can deal with this 3D data. So this is a very large area, no, not large area, the deep 3D G-stack imaging data obtained from the cleared brain. And then we can make this 3D model, 3D rendering, and then see and observe the 3D structure of the tissue. And then again, we can do this 3D imaging at different time point to make this 4D data set. And then after that, we can nicely see how the fluorescent cells moves around inside the 3D space in the live tissue, in the live animal model, so which we call 4D data set. And then recently, we also making this robotic confocal and two-photon imaging system. So we miniaturized our microscope and attached it to this articulating robot arm. And then this robot arm can be controlled, can cooperate with the human. So we can put our microscope in anywhere, in any directions. Okay. So this is a very nice system for the very large animals, such as non-human primate or pig or even dog. Okay. And after replacing our microscope, this robotic arm can be remotely controlled in micrometer scale. And then, so that's why we can obtain these very high resolution images using our microscope in anywhere in a large animal model. Okay, so to summarize, intravital microscopy is a technique for the 3D cellular level imaging of the live mouse model. So we can image many different internal organs in a live animal model. And then we can do dynamic 3D imaging of various cellular level processes and dynamics in the live animal model, as you can see in these examples. And also for the drug development, this intravital microscopy can enable a very direct in vivo drug imaging analysis to monitor the drug delivery to the target cell, and then to assess the drug efficacy in the target cell, and then to validate the mode of actions in a live animal model in vivo. Okay, so if you are interested in this technology, please visit our webpage, www.ibeamtech.com, or just send us email to information, directly send us email to us through information@ibeamtech.com, or please contact the ATCG in Hong Kong. And then we have also some solution for the intravital imaging service as well. All right, so thank you very much for your attentions, and then I will be happy to answer any questions.
Okay. Thank you, Dr. Kim, to give us a wonderful presentation. Now we will go to the Q&A session. The question one is, what challenge arise when labeling drugs or cells with fluorescent probes?
Oh, can you repeat the question one more time? Sorry.
What challenge arise when labeling drugs or cells with fluorescent probes?
Well, so we have a labeling kit to label the antibody with the fluorophores. So yeah, so if you want to label the specific cells in a live animal model, so we need to then if the cell has a specific surface marker, then we can use antibody to specifically label those cells in a live animal model. So what we need to do is we just prepare the antibody and fluorophore, and then using our conjugation kit, we can conjugate the antibody and fluorophore. And then we can either intravenously inject those antibody conjugate or local injection to the target tissue to label the specific cell for the fluorescence imaging using our microscope in a live animal model. Okay, and then let me answer the questions in Q&A window. So the first question was, are there efforts to combine I-beam with other technology like optogenetics or CRISPR to study gene expression or neuronal activity in real time? Yes, there are. Of course, there is some effort. So we can apply our intravital microscopy system for optogenetic mouse model. Or any kind of gene editing system, and then to monitor the neuronal activity in a live mouse model. So I during my presentation, I just showed only a few examples in the brain. But they're using this cranial imaging window technique, which this cranial imaging window. And then this is CIC window technique. We can nicely see the neurons and then other type of cells in the brain. And then also, as you asked, we can combine the optogenetic tool to monitor some genetic expressions and then the activity in the brain. Yeah, so I hope I can answer your questions. Okay, the next question is, what challenges arise when labeling drugs or cell with the press probe? Okay. So as long as the one thing for the labeling the drug is the process probe has its own molecular size, typically 300 to 600 Dalton. So if your drug is too small, about the small molecule drugs, it's very difficult to label those small molecule drugs with the process probe. But other larger molecules such as antibody or peptide or exosome, nanoparticle, or even the cell and gene therapy drugs, they are relatively much easier to label with the process probes. So normally we can use NHS, amine and then amine reactions to specifically label the peptide or the antibody drugs. And then we can also provide those kind of labeling kit as well. Okay, the next question is, what methods do you use to quantify cellular behaviors such as migration rate, proliferation, or apoptosis for I-beam imaging? And then how do you ensure the reproducibility and accuracy of this measurement? Okay, for visualizations, we normally use our I-beam studio softwares. But for quantification, we normally use commercial software such as Imaris or ImageJs for the detailed quantifications. So such as migration, proliferation, apoptosis, or cell-to-cell interaction, or cell morphology, all those cellular parameters can be quantified by using ImageJ or Imaris. And then our microscope, the imaging data is compatible with those, Imaris, compatible with the analysis software such as Imaris and ImageJs. And then the reproducibility, in case of reproducibility, it's more a kind of a matter of the analysis software. But anyway, normally for in vivo intravital imaging, we normally use the number of mice to at least 3 to 5 for the statistical analysis. And then another good thing of intravital imaging is we have a very large volume of tissues. So we can take many images and multiple different sites. And then by doing that, we can increase and we can ensure more accuracy in our quantification in terms of statistical analysis. Okay, and then next question is, are there any protocols for imaging mouse embryos? Okay, so mouse embryo is actually very tricky organs we can images. But there, but there are some reports. There are some reports about the live mouse embryo imaging. So definitely we can try to image the pregnant mouse model. So in this example, as you can see, we have been able to image the placenta in the pregnant mouse model. So the protocol further need maybe some modification, but there is some report about the mouse embryo imaging using two-photon microscope. So we can start from there and then further optimize the protocols. Okay, and then the last question is, how deep can you see the organ in an I-beam microscope? Okay, so as I told you, our microscope is capable of both two-photon and confocal imaging. And then using our microscope, we can normally, so with the confocal imaging mode, we can normally penetrate up to 100 micrometer from the organ surface. It depends on the type of organ and the wavelengths of the excitation wavelengths, but we can expect to 50 to 100 micrometer using confocal imaging mode. Using two-photon imaging mode, we can penetrate deeper. So using two-photon imaging mode, we can expect up to 200 to 500 micrometer in certain type of tissue. Again, it also depending on the type of tissue. So in case of a brain, we can easily penetrate up to more than 400 micrometer because the brain is relatively transparent. So for example, so this example is more than 400 micrometer from the surface. But in other organs such as muscle and skin, normally we expect 200 micrometer by using two-photon imaging mode. So yeah, so that's the rough estimation. With the confocal, 50 to 100. With the two-photon, we normally expect 200 to 500 micrometer, depending on the organs. All right, so thank you very much. I think I answered all the questions in the Q&A.
Okay, thank you. Thank you all for your question. The webinar recording will be available soon on YouTube channel. Actually, you can, for any further inquiry, you can just contact ATCG Limited or send an email that's Dr. Kim mentioned in the PPT by information@imibtech.com. On the other hand, the IM technology now conducting the demos at research institutes and universities around the world. And they also provide custom virtual demos tailored to each research interest. Thank you again for joining us today, and we are looking forward to see you in the next webinar. Thank you very much.
Thank you.